Okay, hello everyone. Thank you for joining this seminar on Bulkhead Permeable Reactive Barriers, or PRBs, for groundwater nitrate remediation. I'm Molly Graffam, and I'm a PhD candidate from the School of Marine and Atmospheric Sciences. Before I get into the presentation, I'd like to acknowledge others who are involved in this work who have really made it possible. From the center, there's Chris Gobler, Frank Russo, Stuart Waugh, and my advisor, Nils Volkenborn. I'd also like to recognize our collaborators from Cornell Cooperative Extension, who have made a lot of this work possible. And the Center for Clean Water Technology receives funding from New York State DEC. Some of the work I'll be talking about today was funded through the Water Quality Improvement Project program, and some was funded through the Town of Southampton Community Preservation Fund. I've also received a stipend from the STRIDE program, which is funded through the National Science Foundation. I'll start by going through the basics about permeable reactive barriers and some of the frequently asked questions that people have. I hope to answer some of these questions for you. And the last half of the presentation will be about two specific Bulkhead PRB projects that are ongoing. So first, what is a PRB and how does it work? Well, groundwater nitrogen has various sources, including wastewater, fertilizers, and products of the fossil fuel combustion that end up in our precipitation. And groundwater moves towards the shoreline and eventually vents into the surface water in a process called submarine groundwater discharge. This groundwater discharge has been identified as a major source of nitrogen to surface waters. Surface water nitrogen pollution is directly related to the occurrence of algae blooms and other water quality problems. Innovative solutions are needed to reduce nitrogen loading to coastal water bodies, and a promising strategy is permeable reactive barriers installed close to the shoreline to treat contaminated groundwater before it discharges into surface waters. The unique thing about PRBs is that they can treat nitrogen derived from all of the major sources, including septic-derived, agriculture fertilizers, and atmospheric deposition. As long as it's in the form called nitrate when it reaches the barrier, it can treat nitrogen from all of those sources. Permeable reactive barriers are a type of passive groundwater treatment. The PRB intercepts contaminated water, and through a series of microbially mediated reactions, the water is treated. The microbes that I'm talking about all exist naturally in the soil, so the key is to engineer the system to provide the right conditions so that microbes can do what they do naturally. And the reactive media within the barrier is typically wood chips mixed with either gravel or sand. This coarse media is more permeable than surrounding soil, which promotes water flow through the treatment zone. There is extensive research on PRBs, and they have been successfully used for groundwater nitrate remediation. There are several forms of nitrogen, and under certain circumstances, some forms are considered contaminants, while others are considered completely safe. Ammonium and nitrate are nutrients for all living things, and when they're found in excessively high concentrations, they're considered drinking water and surface water contaminants. The PRB is particularly designed to treat nitrate. So it first relies on microbes converting ammonium to nitrate before that nitrogen-rich water even reaches the barrier. Nitrate must be present for the barrier to work. And then microbes can convert it to an intermediate called nitrous oxide. But we really want the conversion to happen all the way to nitrogen gas, which is shown on the far right side of the reaction, which is completely harmless and makes up 80% of the air we breathe. So when we monitor a PRB, we sample the groundwater upgrading of the barrier, as well as within the barrier and just after the barrier. And the decrease in nitrate concentration is how we calculate removal and assess the PRB performance. One new application is to install these barriers behind perforated marine bulkheads. And just so everyone is aware, in case you're not sure what a bulkhead is, we are defining it as just a retaining wall installed along the shoreline that prevents erosion, and it protects human habitation against flooding or wave action. Here is an idealized representation of a bulkhead PRB and the major chemical reactions that can occur. On the left-hand side is nitrate-rich groundwater, and the brown box represents the PRB. The black dotted line on the right-hand side represents the perforated bulkhead, and the brown small-looking shapes in the middle is representing the wood chips, which are a source of carbon. The carbon is broken down and consumed by microbes over time, and this process drives down the oxygen concentration. That's when microbes called denitrifiers kick into action and start using nitrate and converting it into nitrogen gas through a reaction called denitrification. The PRB is engineered to perform those first two reactions really well. However, if nitrate is completely removed, there are microbes that can start to shift into performing other reactions that are shown here, and these can lead to some unwanted gas formation and release. So the design process is always aimed at minimizing those unwanted reactions. People often wonder how long a PRB will last after it's installed. This is data from a PRB treating groundwater contaminated with septic waste. The PRB was functioning well for 15 years, and it's the longest data set available on a denitrifying PRB. These scientists measured organic carbon content and found that 80% of carbon was still there after 15 years, so less than 1.5% of carbon was used per year. This means that even if only half of the carbon available was able to be used by the microbes, that the PRB would exceed 30 years lifetime without any maintenance. This study, however, was done on a freshwater PRB, and PRBs installed very close to the shoreline, such as those behind bulkheads, may experience some seawater intrusion due to tidally induced water flow. Under these conditions, alternative reactions that I mentioned in the previous slide can occur, and this may decrease the PRB lifetime, but an effective design, as I mentioned, can minimize these reactions. Since bulkhead PRBs can experience seawater intrusion, we did a lab experiment to test whether the presence of seawater impacts nitrogen removal. We had 12 PRB columns and 3 different salinity treatments, which were freshwater, low salinity, and high salinity. And I took samples over the course of 2 weeks, and I found that all treatments removed greater than 95% nitrate on average, and there was no significant negative salinity effect on nitrate removal. We actually observed significantly less methane in saline columns compared to freshwater columns, which was an interesting result that I'll talk about a little bit later. So in the same experiment, we tested this PRB column effluent for the chemical signs of alternative reactions that I mentioned earlier. And we found significantly less methane in saline columns compared to freshwater columns, which suggests that seawater can actually have a protective effect against the release of methane, which is a potent greenhouse gas. We also found that sulfide concentration increased slightly with increasing salinity. And sulfide is that chemical that smells like rotten eggs, and sometimes you can smell it at low tide in marshy areas. We also found that some iron sulfide minerals precipitated, and this decreased the release of free sulfide. So in this experiment, we found that all sulfide values were well below typical marine pore water values. And this was a very promising result. And again, minimizing these alternative reactions is always the goal when designing the PRB. A really important factor is determining a suitable location for a PRB. Denitrifying PRBs will be best suited for areas with high groundwater nitrate concentrations and appreciable groundwater discharge through permeable sediments. Areas with no other natural nutrient removal mechanism will be ideal locations for the PRB. And also, we want to install them near shorelines adjacent to impaired water bodies. Typically, we want to target shorelines with groundwater travel time on the scale of decades, because the PRB will also last for several decades. And Suffolk County has already identified possible areas for advanced treatment and prioritized these areas according to land use, residential density, shallow depth to water, and location of impaired water bodies. This is a great starting point to identify areas that would be suitable for PRBs. It's really important to think about groundwater travel time when considering PRB placement. Groundwater travel times range from anywhere from 0 to 2 years, which is shown here as the pink color, which actually it's less than 10 years is shown in the pink color, all the way up to greater than 1,000 years, which is shown on this map by the green color. And right now, there's a lot of nitrogen being stored in the groundwater as it travels towards the shoreline. So this means that if all residential wastewater infrastructure was updated today, legacy nitrogen would still continue to impact surface water for decades to come. And while Long Island transitions over the next years and replaces failing wastewater infrastructure, the strategic placement of PRBs can provide more immediate water quality relief. Wastewater infrastructure updates are absolutely critical, and we see PRBs as a complementary to this effort since they can be used as a stopgap solution. Locations that fit the criteria of having a nearby impaired water body and decadal groundwater times may also have existing bulkheads, which are promising potential locations for PRBs. We think that installing PRBs during scheduled replacement of existing protective shoreline infrastructure can be an efficient installation strategy because the excavation machinery is already on site, thus reducing machinery mobilization costs. Bulkheads can also be easily perforated to control groundwater flow. PRBs would then add a function to structures which currently provide no environmental benefit. And just to be clear, we're not advocating the idea of installing new bulkheads. We're only proposing PRB installation during replacement or possibly retrofitting existing bulkheads. And I think it would be useful to have a PRB siting tool for Long Island that includes the existing information which prioritized areas for advanced treatment, and to take it a step further to add data that's relevant for PRBs and perhaps even add information about shoreline infrastructure such as bulkheads. This type of tool could help prioritize suitable PRB locations. Cape Cod, which I'm showing here, has already developed a PRB siting tool. And this is a screenshot of the tool. It's basically a map where you can interactively add different layers that show land use and water quality data, among other information. And there's also a layer which shows the location of conceptual PRBs, which are shown here as the red lines. Although land use and water quality data can point us in the right direction of where to install PRBs, ultimately there needs to be localized site characterization to determine whether a site is suitable for a PRB. Site characterization is critical for the design process. It can be quite extensive, and the bullets I've provided here is certainly not an exhaustive list. But these are some of the things that we think about during site characterization. And the take-home message here is that not all potential sites will be suitable for a PRB, and each PRB will need a site-specific design. And there's a reference here that I provided that goes into a lot of detail about the site characterization of some potential PRB locations in Cape Cod. The design process involves determining the PRB location and dimensions. Throughout the design process, the goals are to maximize nitrogen removal. At the same time, we want to avoid complete nitrate removal to minimize those unwanted alternative reactions that I mentioned earlier. And of course, we want to minimize installation costs whenever possible. Some important design considerations for bulkhead PRBs specifically include the tidal influence on water chemistry and groundwater flow and velocity. Also, the density and size of bulkhead holes is important. We want to make the bulkhead porous without compromising the structural integrity of the bulkhead. The media composition is also an important factor, such as the wood chip type and the wood chip to sand or gravel ratio. Ultimately, we come up with one number for PRB thickness, but a lot goes into figuring out that ideal site-specific number. So, not to get too technical here, but I just wanted to give you an idea of what goes into determining the PRB thickness. So what I'm showing here is water table elevation in the top graph and over time for two different wells, up gradient and down gradient. And to determine the bulkhead PRB thickness, site characterization usually involves these water level loggers in multiple wells at varying distances from the shoreline. The difference in water table elevation between different wells allows us to calculate the hydraulic gradient, or what's called the change in water height divided by the distance between the wells. So that's what I'm calling as delta H here. And near the shoreline, this gradient changes over time, as you can see in the graph, due to tidal influence. This is what causes the submarine groundwater discharge. At high tide, such as in that top graph you can see around 3 p.m., that's 3 a.m. At high tide, the gradient is smaller, so the difference between those 2 points is small. And at low tide, such as between 9 and 12, the gradient is larger, or the difference between those 2 points is larger. With some additional information about the soil characteristics, we can then calculate what speed the groundwater is flowing. And now, if you look at the bottom right-hand graph, you can see the gradient and how it relates to the groundwater speed. At low tide, the speed is higher, and at high tide, the groundwater speed is lower. So you can see there's an inverse relationship between the tide and the groundwater velocity. So once we know the speed that groundwater is flowing over a few tidal cycles, we can plot the data in a different chart called a histogram. And the histogram is on the bottom right-hand side of the slide, and this is showing us what speeds were occurring and how frequently they occurred. Then we can use the most frequent groundwater speeds in the PRB design. It gets a little bit more complicated than that. If it wasn't complicated enough, it gets a little bit more complicated because the wood chip media is actually more permeable than the surrounding soil. So it's important to consider the effect of this high permeability wood chip media, which may draw more water into the media, resulting in increased groundwater speed. And in this diagram, the red box indicates the location of the PRB, where there is high permeability media compared to the surrounding soil. If the permeability of the media within the PRB was the same as the surrounding soil, then you would see just horizontal streamlines. But because of the increased permeability within the media, this results in water being drawn in from below, and you can see these curved streamlines that show water from below moving into the PRB. Another factor that goes into the design is the fact that microbes require low oxygen conditions to denitrify. However, at high groundwater speeds, such as during low tide, a PRB can get inundated with oxygen-rich groundwater. And this reduces the volume of functioning PRB. And in our PRB column experiment, we observed this phenomenon happening. We used an oxygen sensor on the inner wall of a PRB column. And that's what you're seeing in the figure on this slide, is images of oxygen concentration at different water speeds. The water speeds are increasing from left to right. And wherever there is oxygen-rich water, you see white and red colors. So we pumped oxygen-rich water into the columns, and the water flow in this case is from bottom to top. So as water moved through the PRB, oxygen was consumed until there was no oxygen left, and that's shown by the black color. Going from low to high groundwater flow resulted in oxygen-rich water penetrating deeper into the PRB. So you can see that red and white colors moving further into the PRB with increased speed. And this shows that the PRB, the volume capable of removing nitrate was decreased. So this is an important thing to consider to account for the potential loss of functioning PRB volume during periods of high groundwater flow. Another important question is the type of wood chips to use in a PRB, and we think it's important to find the best wood chip type out of the ones that are readily available on Long Island. Cornell Cooperative Extension actually has a shoreline PRB that was installed in 2016 in East Hampton, and it contains treatment cells with 4 different locally sourced wood chip types, including pine, oak, a mixture of pine and oak, and a mixture of maple cherry. Cornell harvested those aged wood chips from the field PRB and brought them to Stony Brook, where now we're conducting a lab experiment with those aged wood chips. That's what you see in the photo. The goal of this experiment is to relate greenhouse gas concentrations and water residence time and nitrogen removal to the wood chip type. In this experiment, the columns received influent with 25 milligrams per liter nitrate, which is shown in the red dashed line, and the water residence time in the columns was about 1 day. This preliminary data suggests that all wood chip types were functioning because there's a difference between the influent and effluent nitrogen values, but there is a trend where pine wood chips removed the least amount of nitrogen, and the maple cherry mixture is outperforming the other wood chip types. This is a very interesting result, and this is consistent with Cornell's field results, which also showed that the maple cherry mixture is performing the best in the field. After all this information about what goes into siting and designing a PRB, you might be wondering, are they cost effective? Some studies have put a price tag on nitrogen removal, and one Florida study estimated the cost of nitrogen removal is $23 to $51 per pound of nitrogen removed. There are also costs associated with ecosystem damage, and these can be considered as the cost associated with doing nothing and letting the problem get worse. If we take a reasonable estimate of how much nitrogen we can remove with a 100-foot bulkhead PRB, then we can calculate a very preliminary cost comparison for the 30-year expected lifetime of the PRB. And although the PRB is expensive upfront, this preliminary cost comparison suggests that it can be a cost-effective approach. So now I'd like to switch gears and talk about the two specific bulkhead PRB projects that are ongoing. The first project is the Hampton Hills Association Bulkhead PRB. This PRB site was chosen based on land use data, groundwater modeling, and also some previously known groundwater nitrate values. The area shown in the top left corner by the small red box is basically what's being shown in the zoomed in larger map. This is a medium density residential area, and groundwater travel times were used from the Task 2A subwatershed model to make this map. And you can see in the little yellow mark indicates the pilot PRB location. So a bulkhead PRB test cell was installed in 2015 by Hampton Hills Association in consultation with Cornell Cooperative Extension. The pilot was approximately 2 meters wide by 2 meters deep and 1 meter thick. A grid of holes was drilled into the below ground portion of the bulkhead sheets to guide water flow through the PRB media. And at this pilot, a perforated pipe was installed in the center of the PRB. So on the figure, you'll see PVC pipe. And this is where we deployed sensors to monitor the chemical and physical conditions within the PRB. Here are the results from the data logger that was deployed in the PRB test cell. And this is a bit of a complicated figure, but the main point is that the test cell was strongly influenced by tides. As you can see, the gray shaded area shows the water level changes over time. And the salinities shown in the green color, those salinities range from 0 to 11 parts per thousand. The DO, which is shown by the blue color, or the dissolved oxygen, ranged from 0 to about 5 milligrams per liter. But conditions in the center of the PRB remained anoxic or had no oxygen during 22% of the time, and the conditions were hypoxic or low oxygen during 73% of the time. So this data suggested that the conditions were supporting denitrification. And what this data really showed us is that the test cell was a dynamic system with bidirectional water flow and variable flow rates through the tidal cycle. We also monitored nitrogen in the PRB test cell. Here we have the y-axis is nitrate concentration, and the secondary y-axis is tide height. The x-axis is time. So I'm showing nitrogen concentrations in the groundwater and within the PRB over time. The groundwater is shown by the blue circles. So there was about 5 milligrams per liter on average in the groundwater, and it varied a bit over the tidal cycle. The green squares show partially treated water from within the PRB, and the black triangles are showing fully treated water where samples were below the detection limit at all time points. And so this was a very promising result. And most of this preliminary work took place in 2017. Then in 2018, the Hampton Hills Association was awarded a CPF grant from the town of Southampton to install a 100-foot bulkhead PRB based on those promising results from the test cell. And in 2019, paperwork and contracts were finalized and Cornell began site characterization. Then in 2020, the site characterization and PRB design were finalized and construction and installation occurred. We were able to get this done amid coronavirus, but we were doing it very safely, all wearing masks and socially distanced. So you can see in the photo from this site where we had an educational sign, and in the back of that photo is the massive pile of wood chips. Here's a top view of the study area with the yellow line showing the length of bulkhead, which was recently replaced and now has the PRB behind it. This section is approximately 120 feet long, and the PRB itself is not a continuous trench, but instead is broken into 12 treatment cells, and there's also a regular trench section on the far right-hand side. So when Cornell and Stony Brook groups were thinking about the design, we saw it as an opportunity to gain useful information about PRB thickness and PRB type in order to make better future designs. So there are 4 different types that we settled on: the control, 1 meter thick, 2 meter thick, and a new PRB column type design with staggered columns filled with wood chips. And there were 3 replicates of each type so that we can ultimately make meaningful statistical claims about the PRB performance. And the controls had no wood chips, so the additional trench area shown on the far right was added to make sure there was at least 100 feet of functioning bulkhead PRB, which is what we proposed to the Southampton CPF board. So here's a photo from the installation so you can see behind the scenes what was happening. And here is the 2 different thicknesses, the 1 meter and 2 meter thick PRBs. You can see there was a permanent bulkhead installed between each of the 12 treatment cells to ensure that there was no lateral water flow or exchange of water between the treatments. And this would, if there was lateral water flow, that would make it harder to distinguish the effects of each treatment. So these bulkhead returns were installed, and they did increase the cost, but we felt it was worth it for the scientifically rigorous test. And future installations may not require those bulkhead returns, but they also added some structural stability. The bulkhead that you see in the picture on the landward side was temporary, and that was removed after the treatments were installed. Here's another photo from the installation, and this is showing the PRB column setup. You can see from the photo multiple rows of PRB columns and the white pipe sticking out of the center. I just wanted to point that out here because this is where we were able to add continuous loggers, again, so that we could collect a lot of data. And there were also multiple sampling ports installed in each PRB type. Here's a photo from after the PRB installation. So you can see the beach area looks totally normal, and most people don't realize there's a PRB along the bulkhead, although we do have that informational, educational sign for people who are interested in learning more about the project. So just to go into some of the preliminary data from this site, I mentioned that the white pipes in the middle of each treatment cell were for data logger deployment. Here is some of that preliminary data from a few of the treatment cells. Water level in this graph is on the y-axis and time is on the x-axis. And here the blue line shows the tide water level, and the tide was stationed about 40 feet into the bay. This data set is for for 24 hours that I'm showing, and you can see the semi-diurnal tidal pattern, which means there are 2 high tides and 2 low tides per day. The black, red, and green lines show the water level in the control, 1 meter thick, and column setup. You can see the maximum and minimum water levels in the PRBs slightly lag compared to the tide gauge. And this makes sense because they're onshore, and whereas the tide is offshore, so it's experiencing the tide effects before it reaches the PRBs. This also shows that the different treatments, these 3 treatments, look very similar. So it looks like they're performing hydraulically similarly. And the loggers were, it looks like they were deployed at similar depths. This really confirms that we installed the pipes correctly and we installed the treatments in a similar depth horizon. So this is all confirming that the installation was successful and that they're hydraulically functioning. In addition to the continuous monitoring, Cornell also started to sample the ports that are up gradient and within the PRBs. However, there's not a full data set yet, so I'm showing data from 1 treatment cell of each PRB type. On the left is a graph of conductivity, which is similar to salinity in that it's a measure of the amount of salt and ions present in water. And the black color here shows the up gradient data, and the up gradient sampling ports were those on the landward side of the PRB. Whereas the white color shows the down gradient sampling port, which is just after the PRB, and this is representing treated water. So in regards to the conductivity, the 1 meter thick and the column setup look very similar to the control, and they have very fresh water samples. The 2 meter thick PRB has slightly higher conductivity than the other PRB types. The down gradient samples specifically have higher salinity than the other PRB types. And this could be suggesting that there is some salty water getting into the PRB. But again, the data is all showing that the water is very fresh, because actually 2,000 microsiemens is generally considered fresh water. So this is all really promising, because it's showing that groundwater is flowing through the PRB, and we're getting not very much seawater flowing into the PRB. And on the right side, the figure is showing dissolved oxygen in the different PRB types. And you can see there's a decrease in dissolved oxygen between the up gradient and down gradient ports for all of the PRB types except the control. This makes sense because there's no wood chips in the control, and so we don't expect to see any difference between up gradient and down gradient samples. But it makes sense that the dissolved oxygen is decreasing in those PRBs, because that's exactly what they're designed to do. They're designed to draw down the oxygen and then start allowing the microbes to use up nitrate. Here's some of the preliminary nitrate data collected from 1 of each type of treatment. There are a few caveats that I'd like to mention first, which is that this data was collected with a nitrate sensor, which has a method detection limit in the range of 1 to 3 milligrams per liter. And compared to the traditional way of measuring nitrate in the lab with a nutrient autoanalyzer, that detection limit can get to less than 0.1 milligram per liter. So this sensor has a higher detection limit than the lab equipment that would typically be used. The sensor is great for screening purposes though, since it's portable and it's fairly inexpensive. And however, when we're dealing with nitrate concentrations below 5 milligrams per liter, we really want that low detection limit And ultimately, we will need to analyze these samples in the lab. Another important point is that the nitrate sensor can only measure freshwater samples. So if we do encounter samples with greater than 2,000 microsiemen conductivity, the nitrate sensor can't be used on those samples. So although the data is preliminary, the results shown here suggest that the PRB is functioning and removing nitrate. You can see a decrease between the up gradient and down gradient ports for the 3 different PRB types, while in the control, there is no decrease, and actually there's a slight increase in nitrate between the up gradient and down gradient ports. So there's no removal happening in the control. And just so you're aware, the ND means not detected. No nitrate was detected in the down gradient samples of 2 of those PRB types. This is very promising results. Of course, we have next steps. The initial round of sampling will occur soon, and these samples will not be analyzed with the sensor. They'll be analyzed in an ELAP laboratory for nitrate and for other nitrogen components. The resources are available to do the minimum required by the contract to evaluate the PRB performance. But we would like to use this opportunity to expand our understanding of bulkhead PRBs. So the plan is to secure more funding for long-term monitoring and to study the PRB in a scientifically rigorous way, since it is already nicely set up with the replication of each treatment type. It's a great opportunity for research. So that concludes the portion about the Hampton Bays Bulkhead PRB. And I just want to briefly cover the work that has happened so far at the Davis Park Marina site. This project is still in the site characterization phase, and results are shown from one sampling event in late 2019. The yellow line here highlights the bulkhead area, which is the proposed location for a PRB. And during site characterization, 6 wells were installed on the landward side of the bulkhead, shown here by the blue dots. There were multiple depths sampled at each location, so the numbers that you see next to the blue dots are for the different depths, and they were 10, 15, and 20 feet below the surface. Oxygen values at this site ranged from 0.53 milligrams to 5.69 milligrams per liter, and low oxygen values were observed particularly in the center well closest to the bulkhead, which was near the restroom area. This is the same map, but now instead of oxygen, the data is nitrate. And the nitrate values ranged from below detection to 3.267 milligrams per liter. However, most of the nitrate values were less than 1. I'm not showing it here, but during the initial sampling round, there was also high ammonium found in the well where there was low DO and low nitrate. That's the middle well closest to the bulkhead. This suggests that there is septic-derived nitrogen contributing to the nitrogen pollution problem, but there is not enough oxygen available to convert the ammonium to nitrate. And there will likely be another sampling effort in a different season to see if the nitrate values have changed. So in summary, installation of denitrifying PRBs during replacement of bulkheads is a promising and efficient approach to reduce nitrogen input to coastal waters. And PRBs are unique in that they can remove all forms of groundwater nitrate regardless of source. So they can remove nitrogen from fertilizers, septic, and atmospheric deposition as long as that nitrogen shows up to the barrier in the form of nitrate. Nitrogen removal performance is not negatively impacted by saline conditions, but saltwater intrusion can be minimized when the PRB is designed properly. The Hampton Hills Association PRB is now fully installed and preliminary data suggests it's functioning well, and we are taking the next step soon on getting the full sampling effort completed. The Davis Park Marina PRB is still in the site characterization phase and needs more site assessment to determine if the location is suitable. So with that, I'd like to say thank you again to all the collaborators who made this work possible, and to our funding sources and everyone involved who participated in some way. And here's the slide showing the references that were cited in this presentation if you're interested in learning more. I'd also like to mention that if you want to hear more about the different technologies that the center is pursuing to tackle the nitrogen problem, there's another upcoming talk on September 16th at 6 p.m. by Chris Gobler called The Future Is Now. And with that, I'm ready to take any questions if you have any for me. Thank you.
Molly, this is Scott Horsley. Can I ask a question?
Sure.
Thank you. Yeah, I'm sorry I joined a little bit late, but I'm interested to know if you could explain wood chip diameter characteristics, and I don't know if you did any permeability calculations or measurements.
The wood chip diameter is important. There has been research on different diameters. Specifically, sawdust has been used in some PRBs, and using sawdust creates more surface area that's available for microbes to start breaking down that carbon. So I think the sawdust has been used successfully, but we typically have been using wood chips just because we want the carbon source to last as long as possible. And so far the wood chips have been performing really well. So the diameters range, I mean, I think the diameters have a wide variety of range because it's just like chopped pieces. We haven't really been sorting it necessarily for specific size ranges, but we also do conduct permeability tests in the lab, and they're a bit challenging just because the permeability is actually so high that it's hard to even be able to detect a difference in pressure head because just the water is flowing so quickly through the wood chip media. But there has also been literature calculating the permeability, so that's available if you're interested in hearing more about that.
Thank you.
I think I see another question. Have you or your colleagues sampled near the tops and bottoms of PRB cells to address any vertical components of groundwater flow? So that's a great question, and yes, in our PRB column experiment, we definitely noticed that the flow is not just in the horizontal direction because there's a lot of tortuosity. The flow can be moving in the vertical direction as well as in the horizontal direction. So we saw that in the PRB column experiment, and then in this most recent Hampton Hills Association PRB, we have a few sampling ports that are installed at different depths within the PRB to address that vertical component question as well. So I think we're poised to start to get at that question.
Molly?
Yes.
This is Legislator Krupski. I think that was a very, very good, well-designed experiment, and it's very controlled the way it was set up and everything. Is there any in the future? Is there any you know, what are the plans for the future future use of this? It's pretty simple chemistry, and it has great utility. There are an awful lot of bulkheads being replaced every day on Long Island. but the way this was designed, it's obviously fairly complicated in trying to determine which soil types are which and the flow rates, versus having a contractor come in with a bulkhead replacement permit and installing this as part of a sort of a routine installation, instead of each one being, I don't want to say over-designed, but really, I know you put a lot of work into the design of this, but so is there any hope of having this simplified to make it a little bit more utilitarian for a marine contractor so it could be permitted on a regular basis?
Yes, I think if going back to the slide that showed the 12 treatment cells plus an additional section on the far right-hand side that was just like a regular trench section, that regular trench section is kind of exactly what you're talking about. It wasn't as heavily designed as the 12 treatment cells. And when the contractors were installing that section, it went really smoothly. It was pretty fast because they were getting to be more experts at doing this. So I think there's definitely, we're moving in the direction of getting it to be a more simple process that the contractors can just get these bulkhead PRBs installed without all of the extra work that went into making this an experiment type. But we are going to learn a lot of information from this experiment and then be able to use that and put it in a report so that it can be used in the future.
Thank you. Appreciate that.
So there's a couple more questions. Can you go over the math for the nitrogen removal rates and how that relates to DO and nitrogen levels? So I think in this case, we just showed influent and effluent nitrogen values. So there wasn't really a rate calculated from that yet. But when we do calculate a rate, we're going to be taking into consideration the groundwater flow, so the speed of groundwater moving through the PRB, the amount of water that's treated, and then once we use the difference in concentration between the upgradient and downgradient ports and have that information in combination with the estimates for how much water is being treated per day, that's when really we can calculate a more complete nitrate removal rate, like on a yearly scale. So I think for the Hampton Bays bulkhead PRB, we're not at that level yet for the 100-foot section. We did calculate some removal rates in our PRB column experiment, and we found that for different hydraulic loading rates, there was actually pretty consistent nitrogen removal. So actually, it was an interesting result because the nitrogen concentration and the loading rate were kind of, and the salinity were independent of nitrogen removal. Basically, we calculated a pretty consistent nitrogen removal rate for different influent concentrations and different hydraulic retention times. So I think we've started to address that question in our lab work, and then we'll be addressing it in the field when we have more data. And then another question is, could the secondary bulkhead behind the primary one be eliminated? I think that question refers to the photo that I showed where there was a bulkhead behind the, like on the upgradient side of the treatments. That bulkhead was temporary. That was only there to allow the machinery to put the treatments in, and then the bulkhead behind the treatment was just removed. So I think that answers your question, that it's eliminated. It was temporary. It was just there for the installation purposes. The bulkheads that are perpendicular to the primary bulkhead, those were bulkhead returns that were used to prevent lateral water flow between the treatment cells. And that was, again, because we were kind of over designing this. We wanted to make sure that the treatments were individual sections and that there was no cross flow. So those bulkhead returns may not be necessary when you're not doing this type of experiment. Another question is, can you describe the column treatment in a little more detail? What was the diameter of the columns? How far apart were they placed? What was the soil medium between them? Those are great questions. The diameter of the columns was 10 inches, I believe, and there were 14 PRB columns. So those columns were placed in an area that was the same size as the 2 meter thick PRB. So we basically had the same size as the 2 meter thick PRB, but we used less wood chip media because we had them in these column shapes. And the media that was between the columns was just the natural sand. So when the contractors were filling back in the area to just backfill everything, they just had dumped the sand on top of that, and then we're assuming that that sand filled in those spaces between the columns. And I think we saw that the column setup was performing hydraulically similar to the control setup and the other PRB types. So I think it's reasonable to say that those spaces between the columns got filled in with sand, and that's what happened there. Is there a concern regarding erosion of the PRB media through the holes in the bulkhead, and especially when considering higher than normal groundwater velocity? So there really is not a concern that the PRB media itself will go through those bulkhead holes. The media was kind of enclosed, so it can't get out of the bulkhead really, because it's just kind of stationed behind the bulkhead and the wood chips aren't moving. The carbon itself that kind of dissolves from the wood chips is somewhat mobile within the PRB, but the wood chips themselves are not going to go through the holes in the bulkhead. How deep are the columns is another question. I think at this site, there was about 10 feet of treatment. So about 10 feet worth of wood chips was installed below the groundwater level, if that makes sense. I think that's all of the questions. So again, there was a lot of questions. I'm really glad that everyone was kind of interested in this topic, and hopefully I didn't get too technical on you, but thank you again for joining, and I hope you all have a great rest of your day.